Today I'll be presenting analysis of total oil and protein percentages from 2018 to 2020. Peanuts provide an important source of plant-based protein as well as other vital macro and micronutrients. For the past 10 years, the Biochemistry Research Lab at Lubbock Christian University has conducted crop analyses in conjunction with Birdsong peanuts. Given our experience from 10 years of crop projects, we have noted the importance of oil amount in relationship to maturity and subsequent quality of the peanut. As the plant matures, the amount of oil also increases to potentially more than 50% of the total seed mass. This made us question, if the oil content can increase with maturity, what component might decrease in order to provide the raw materials needed to build additional lipids. Two past studies noted the correlation between total oil and protein percentage in specific genotypes. Dumas et al. In 1990 reported a high correlation and inverse relationship between total oil and protein, negative 0.38 to negative 0.71, at different environmental conditions. Wang et al. In 2016 also identified the inverse relationship between oil and protein for the U.S. mini-core collection, but the correlation was not as high, negative 20.23. In other words, both studies observed that as oil increased, protein decreased. Given this information, would the same relationship be observed in different genotypes, growing regions, and environmental conditions for cultivated production samples? To answer this research question, we conducted a 3-year study with southeastern runners during the U.S. peanut harvest. Samples were gathered at 13 or 14 different buying points in Georgia, Alabama, and Florida during the 2018 to 2020 crop years. About 25 samples each year from both large and small subsets were analyzed for total oil and protein percentage for a total of 77 samples over the 3-year period. The samples were randomly gathered with no selection by genotype or specific growing conditions. Total oil was determined by solvent extraction and gravimetric analysis. Total protein was analyzed by the Dumas method at Medallion Laboratories. The total oil percentage was not significantly different than the 8-year average for the southeastern growing region as shown in Figure 1. Percent average and ranges for oil and protein content by crop year are summarized on tables 1 through 3. Oil and protein correlations for each crop year are illustrated in Figures 2 through 4. Not accounting for seed size, length of growing season, or environmental conditions, we did observe an inverse relationship for oil and protein percentage. The correlation was not as high as the Duvetti report from 1990, nor as low, on average, as the 2016 report by Wang. The previous studies represent specific experiments grouped by genotype. Also, it should be mentioned that this study did not control for seed size. Rather, samples were segregated into large and small subsets. In the previous studies referenced, seed size was measured and found to be negatively correlated with protein content. Given the significance of total protein to consumer nutrition, the timing of harvest may be managed to ensure balance between these important quality concerns. Preliminarily, we sought to understand how the macronutrients change with maturity in production samples. Questions for future studies include investigating fatty acid composition, specifically oleic and linoleic acid content, also reported to impact total oil and protein by Wang et al. In 2016. Given the importance of these fatty acids related to variability in normal oleate versus high oleate genotypes, it would be advantageous for the industry to have more information on the nutritive components of peanuts. The authors would like to thank Birdsong Peanuts and the J.M. Smucker Company for providing funding for this project. We would also like to thank Dr. Darlene Cowart and Dr. Chris Leibold for their help conducting the study.